So you would think that this is a really good place for looking at a local translation. However, for the ASAM, a lot of time people initially did not pay attention to it. They more pay attention to the ASAM of gross cone during the early development. Here also showing the local translation can determine whether the gross cone to turn to specific neurotrophic signaling. Okay, but why people exclude this presynaptic mature axonal terminal? So that's what I so-called the dark era. Okay, so the first reason is because they didn't see a polyribosome in the axonal terminal in EM, and I don't know how good you are looking at the EM graph. I cannot. I'm pretty bad. So this I show you. They show the dendrite. Okay, where is the ribosome exists? It's things like this, things like this, things like this. Right. This is so-called a polyribosome, and they didn't find this kind of structure in the axon. But remember, axon is pretty tiny structure, probably even thinner than the dendrite. So you probably don't find it. But recent study, people use more advanced image system. They do detect there's a ribosome in the axon, but often they exist as a monosome. Indicate they probably just have one ribosome on one transcript, and that's sufficient to synthesize the peptide. You don't really need a lot of polyribosome on RNA to synthesize peptide. So basically, that's one of the reasons, so kind of hampered the field for a long time not investigating the axonal terminal. That's one of the reasons. And second reason of all the synaptosome, for example, we're good on doing the synaptosome preparation, but the thing is the pre-post is not so easy to separate. In the mature synapse, there's a lot of cell adhesion molecule. They're kind of connect pre and post. So it's not something you can just separate these two compartments. So in recent study, a lot of study indicate there's a mitochondrial or ribosomal related transcript. It's in this compartment. But how do I know this compartment is in the both or just one part of the synapses? Okay. And the third reason, of course, even when I do the neuron culture, it's easy for me to trace the synaptic spine, but it's hard to trace this axon in the mature neuron where a lot of synapses form in cultures. So that's why a lot of study people done the axonal growth cone. Of course, recent there's a lot of advanced technology. One of it is like this one probably. People claim they can grow the neuron on the filter, and the filter has a pore size and only allow dendrite or an axon to go through. And you can isolate this so-called process compartment. Or more precisely, you can use a microfluid device. They do allow only the axon to go across the channel to the other side, so you can isolate this material. So basically, all this study kind of tell you if you isolate this axonal part from the culture cells, culture neuron, this is culture primary neuron, they do find there's a unique transcriptome, indicate there's an RNA in there, but kind of no indication what they're doing there for. And more cool studies done in the vivo, and this one is still done during the early development. They basically, I think it's implanted cells or electroporation cells. Let the cell express nuclear GFP or membrane RP. And the upper layer of the cortical neuron, they will project their axon to the other side of the cortex. So they isolate this part as the axonal compartment and isolate this part as the so-called somatic compartment. And they can run through the flow and isolate so-called growth cone rich axon. And they also do proteomic. And I think the whole study also find basically mTOR dependent. I think mTOR is very important kinase for protein synthesis. So they indicated mTOR is very important for regular translation. So for me, kind of feel, well, still is regular translation, but there's no indication in which protein has to be synthesizing the axon to drive axon-specific function. Okay. And this study is also done by Aaron Schumann. Both of studies are done by them. And one is by, what I heard from her talk, probably there were thousands of the hippocampus slides, and just the postdoc just isolate so-called cell body and so-called dendritic layer. Of course, this dendritic layer also contains some axonal projection from the CA3 neuron, and they can do so-called ribose-seq or so-called RNA. Here I briefly explain the ribose-seq. So the idea is when RNA is bound by the ribosome, if you lock this ribosome on the RNA, and then you can do the RNA digestion. So the one being bound by the ribosome, this part of the fragment can be preserved. And now you can do the next generation sequence. And finally, you can derive with so-called translatal profile. So when this data, although it's looking at RNA, but kind of indicate what's the translation status of the RNA. And this is just a regular transcriptome indicate the amount of the RNA. And this is the other study, but I want to emphasize from this study, they were able to basically put some of the molecule here and indicate the origin here indicate they're more somatic enriched expression, and the green here more indicate their neuron enriched expression. And you can see a lot of time they assign so-called their transcript. I think it also is kind of biased. For example, I can see some of the transcript involved in the mitochondria that's specifically put in the presynaptic. And some of the transcript involved in translation regulatory factor or initiation factor, they put it in the postsynaptic. Still, we don't know what's the axonal translation. So I guess this is almost, I would say it's my last study with my CPB protein. So that time we tried to address from the CPB2 knockout mice. Is it possible they can regulate local translational in axon? Are they important for learning and memory and long-term synaptic plasticity? So typically the way we did in the lab is we find what protein we want to work on, we generate knockout, and we do superior. And one will be the Morris water maze. And I briefly explain this assay. So basically, we have this water, we put the milk powder in there just to hide the hidden platform here. So the mouse in the water maze, they could not see where the platform is. So we have to guide them and use the special cue around the maze to locate this hidden platform. And after training, they'll get better. And but they have to train like, at least in our case, we train for 4 days. And on the day 5, usually we will remove the hidden platform. And then you test how much time the mice, the mouse, spent around near this target quadrant where the platform used to be. And I think if you have a good memory, you strongly believe this is where the platform is, but you couldn't find it. You were back and forth and swimming around here. So let's use this swimming time, swimming latency in this quadrant. We kind of define what's the consolidated spatial memory. And we also can do electrophysiology and see what happened, what's wrong with their synaptic plasticity by electro recording. And we can culture a neuron and score this postsynaptic synapse morphology, see what's wrong with that. And if there's a phenotype, that's always pretty headache to me. Even though it's the order-only approach, still for me, when we do the genome-wide unbiased study, we always get thousands of targets. But which ones actually can explain our phenotype usually has to put in the educational gaps. Well, actually, it's literature search. If there's nothing in the literature, it's also hard for me to know what people has known before. I sometimes try to use the ChatGPT. Nowadays, a student told me they can put a PDF in the ChatGPT, and they can read the paper for you. But at least yesterday, I was trying to ask, does CPB2 involve this? Some information are right, some information are not. So I think has to be very careful about the information. So this study has been published in 2017, so I just quickly summarize. This is a PhD thesis for my student. So when she uses CPB2 conditional knockout mice, what she found is at the postsynaptic site in the dendrite, the CPB2 regular translation of the protein called GRAPS1, and the GRAPS1 protein is important to help this mPAR receptor. an important neurotransmitter receptor in our system. It's very important to help this mTOR receptor to the synaptic surface. Without the reduced GRAS1 will cause less mTOR receptor on the surface, and that will lead to the reduced LTP and also the impaired spatial memory. Okay. So this is kind of typical study what we found, what's important in the postsynaptic part. And the way we did it is because we crossed with a mice called the CAMK2 This has been used widely in the field because they only pop out your gene postnatally after 3 weeks. So typically, this is time all the neuron is well developed, all the dendrite axon is in place. So we kind of thinking using this pre-mice, we only address synaptogenesis issue. Just look at the synaptic plasticity. And with this CAMK2 pre, we can specifically knock out CAMK2 in the excitatory neuron in the CA1 region. Okay, you can tell, right? From the brown become more pale. But the CA3, the presynaptic terminal, all the proteins still there. So initially when we do this study, when you do the electrophysiology, we definitely find there's a postsynaptic defect, but not a presynaptic. I'm not going to go through the electrophysiology detail, you just have to trust me. So here indicates the postsynaptic, there's a difference, and presynaptic, there's no difference. And this is another looking the neurotransmitter release probability also has no difference in this knockout condition knockout mice. Which is kind of nice control because we don't expect a difference. So because we have this data, we thought it's possible we generate more a global knockout using nesting pre, which you can pop out the CPV2 in all the neuron and also in astrocyte. But nonetheless, it's where the pre we have. So we generate this knockout, now the CA3, CA1 neuron, all the CPV2 is out. When you repeat the electrophysiology recording, we see not only postsynaptic is defective, we also can see presynaptic response of difference, and with all this indication. So this is just kind of basal neurotransmission response. And now we want to move to how about LTP? How about activity-dependent protein synthesis-dependent LTP? So we can give the slice for high-frequency stimulation, and this will induce the CA1 neuron to be depolarized for a long time, for like the last for 3 hours. So in knockout, this is definitely impaired in the CAMK2 pre-conditional knockout mice, because in here we only pop out CPV2 in post, not the pre. But with the nesting pre-mice, now we pop out the protein also in the pre, we can see the significant impairment of this LTP. So this kind of give me the sense maybe our CPV2 regular translation is important in the presynaptic component. Because the way we would jump to look at this issue, I think one of the reasons is because a student I have, I guess later because of COVID or something, so she decided to want to have a kid, so she decided to stay in the lab. And she's pretty good, she has pretty good hand. So I said maybe we can look at very, very hard to look at the axonal translation. Okay. So now the question we see this, we're excited about, yes, it looks like CPV2 regular translation in axon, but which axon RNA are translation or regulate? We really don't know, right? Now, unless you find a target, you demonstrate they're regular by CPV2, no one is going to believe you. So the way we did it with the help from the literature. Okay, so one way we can do is not by doing the transcriptome. Because for people looking for the translational regulation, we expect the target we are interested in, the RNA level does not change. The only change at the protein level. So we don't do the translator because we have done ribostick before, didn't get a good data. So I don't, yeah. So I go back to do one of the way we can look at what's the potential target RNA for CPV2. Is that we can immunoprecipitate the CPV2, and as a control, there's no CPV2, right? Just a control IgG. And you precipitate this, you now check the protein is precipitated by the antibody successfully. Then you can isolate RNA to do the microRNA or do the RNA sequence. So we got like more than 2,000 of the target. And in the same time, the 2019, this study, I think is quite nice, published in the eLife. So they're the first one looking at learning-induced axonal translation difference. So the way they did is they injected AAV, which will express this YP tag L10A. What is this thing? So basically, this is ribosomal protein large subunit 10A, and has been demonstrated this subunit can be successfully incorporated into the ribosome. So when you are pulled down with a YP, you are actually indicating you are pulled down the ribosome. So when you pull down the ribosome, you are also pulled down with a ribosome associated with transcript, indicate they're translating ribosome affinity pure RNA. So they call this assay as a trap. So they do this as a control. They do the YP because when you do this, is that a lot of time there's a lot of background. Just the RNA will stick on the beads, all this kind of reason. So what they did is they put this virus infected. I forgot it's bread or mice. It doesn't matter. In the chamber and then give it a tone and shock. So the rat, probably, I think it's a rat, probably will associate this unpleasant experience with the tone. Okay. So after 90 minutes, presumably after stimulation, there's a lot of protein need to be synthesized to support long-term memory. So they isolate this so-called original soma area, and also in this cortex project to the lateral amygdala is another brain region important for field memory. So they isolate axon terminal from it here, and then they can do transcript study and just put it simply, simplify all this study. We basically take the data, we analyze again with our own way, and then we also take our RIP data and doing some kind of initial screening and make sure the transcript we have contain the CPE element in 3-pound UTR. And both of them all have more than 2,000 transcripts. But when you pause them together, things that were interesting on the transcript they're learning induced, we only have 272. Still is a lot. And then I told my students, say, well, why don't you just narrow down to glutamatergic transmission, since everything we see is on the glutamatergic transmission. So we total have 7, but we eliminate the one in the astrocyte or in the postsynaptic region. So finally, you only have these 3. And one of them is present in both the pre and post, and 2 of them is presynaptic side. And also we know both the transcript encode the protein important for synaptic vesicle function. So that's why we later focus on these 2 proteins. And first thing we did is confirm the protein level. So the SLC70A6 is the gene name, but they encode the protein called the vesicular glutamate transporter. Yeah, so it's a big T2. So you can see either taking the cortex or taking the PIV21 neuron in culture, and this is global. This is from global knockout mice. And we can see a reduction of this protein level, but not synaptotechnic one, not this one. Although both are the target, but only this one changed. And the VGLUT1 is also another important family protein of VGLUT2, and we just confirm it, there's no change. And so the VGLUT2 is downregulated in CBP2, and we also make sure the RNA is the same in the knockout. And this is typically, you would just do a reporter assay. You put a 3-pound UTR fused to the firefly luciferase, and you put this trans fat into the cells. In the presence of the CPV2, we found there's an increase of the protein production, but not the presence of the control EGFP or just the C-terminal domain of the CPV2. So this assay indicates VGLUT2 is translational regulated by CPV2. So we probably can publish a paper by now. But seriously, if you just publish a paper, people will tell you this is another target, and you already published a paper before. So nothing new about nothing new in here. So I told my student, you really have to push to see whether this is locally synthesized in the axon. So when we actually take the VGLUT1, VGLUT2, we do the immunostaining, I was like a shock. You probably can find this data from the literature already, but this is done by my student. So you can see the VGLUT1 is heavily expressed in the cortex and also in the hippocampus in the adult. But the VGLUT2, what we're interested in, is only in the very few region of the hippocampus and few in the CSV region. So it's really hard to think that they can regulate this LTP. So to kind of shift our focus, we kind of determined to take another pathway to record again, which will be the TA pathway. This will be the neuron from entorhinal cortex project to the CA1 neuron, but they project to the more distal